Hello, my name is Professor Hudson, and you are about to view narrated PowerPoint lectures for Chapter 8 on cellular respiration. In the last segment, we covered how do cells obtain energy, what happens during glycolysis, and what happens during cellular respiration. In talking about cellular respiration, we talked about glycolysis, which precedes cellular respiration, and we talked about the transition preparatory reaction, which is where the pyruvate is converted to acetyl coenzyme A. We also talked about how that acetyl coenzyme A enters the Krebs cycle to be further broken down to release electrons and protons, which are gathered in the form of NADH and FADH2. Now that we have primed the system by collecting these electrons and hydrogens, we can do the last stage of cellular respiration, which is the oxidative phosphorylation and electron transport system. During glycolysis and the mitochondrial matrix reactions, such as the preparatory transition reaction and the Krebs cycle, the cell captures many high-energy electrons and carrier molecules. It forms 10 NADH and 2 FADH2 for every glucose molecule that was broken down. These carriers each release 2 electrons into an electron transport chain, which are embedded in the inner mitochondrial membrane. These high-energy electrons jump from molecule to molecule in the electron transport chain, which consists of a series of proteins embedded in the membrane, losing small amounts of energy at each step. This resembles the process that occurs in the thylakoid membranes during photosynthesis. Much of the energy is harnessed to pump hydrogens from the matrix across the inner membrane into the inner membrane space, producing a concentration gradient of protons. The buildup of these protons in the inner membrane space is used to generate ATP during chemiosmosis. At the end of the electron transport chain, the energy depleted electrons are transferred to oxygen, which acts as an electron acceptor. So this is the reason why you actually need oxygen in order to do cellular respiration. As these electrons pass from carrier to carrier in the membrane and generate this current for turning on the ATP synthase that produces the ATP. Eventually, when they reach the end of this chain, these electrons and hydrogen ions must go somewhere. And so the oxygen that we breathe in acts as the final source for collecting these electrons and hydrogens after they've gone through the electron transport chain. 1 water molecule is produced for every 2 electrons that go through the electron transport chain. Without oxygen, electrons would be unable to move through the electron transport chain, and then this in turn would stop hydrogens from being pumped across the inner membrane. And without this ATP gradient, the ATP synthase enzyme would not be able to generate ATP. So the electron transport chain is a series of carriers in the membrane called the cristae, which can accept electrons and hydrogens from NADH and FADH and pass them from one carrier to the next, ending with oxygen, to produce water. All of this forms an energy power source for turning on ATP synthase to produce ATP. This is another view showing more specifically on the process of chemiosmosis. Remember, osmosis is the movement of water from high to low concentration against a membrane. In chemiosmosis, water is not what's moving. In this case, you have protons moving from high to low concentration across a membrane. Protons in the form of hydrogen pumping across a membrane. And so we call it chemiosmosis. As the electron transport chain pumps hydrogens across the inner membrane, it produces a high concentration of hydrogens in the inner membrane space and a low concentration in the matrix. The energy present in this non-uniform hydrogen distribution across the inner membrane is released when the hydrogen ions flow from high to low concentration. As they flow from high to low concentration through the ATP synthase channels, Their movement generates ATP from ADP and phosphate. The newly formed ATP leaves the mitochondria and enters the cytoplasm, where it is providing the energy needed by the cell. Without this continuous recycling, life would cease. It's worth mentioning that people produce, use, and then regenerate the equivalent of their body weight in ATP daily. So we could not survive without a constant supply of ATP. So this slide is just summarizing the overall process. As we stated, the breakdown of carbohydrates begins with glycolysis, and then if oxygen is present, cellular respiration can proceed. Most of the ATP generated from this process occurs during the electron transport chain and the oxidative phosphorylation. This slide shows another view of the ATP generated during this process and where it is generated. Notice during glycolysis we generate 2 ATP. During the citric acid Krebs cycle we generate 2 ATP. We also use ATP during these two processes, so our net yield of ATP is only 4. However, all of the NADH and FADH2 that was generated through the glycolysis and the citric acid cycle is then used to turn on the oxidative phosphorylation and the ATP synthase enzyme in order to generate 32 to 34 ATP, with a total yield of 36 or 38 ATP for every molecule of glucose. Now, while carbohydrates is often used as the example for how cellular respiration can occur, be aware that proteins and fats taken in through our diet can also be broken down through cellular respiration. Remember, we talked about how metabolism is the sum of all of the chemical reactions that occur in the cell. And these chemical reactions can be in the form of catabolism, which is breakdown, or anabolism, which is synthesis. Carbohydrates, proteins, and fat can be used for energy, and their components can be used for synthesis of needed compounds. Both anabolism and catabolism use the same metabolic pool of reactants. This figure is just showing how basically carbohydrates, fats, and proteins can be used as energy sources, and they enter the degradative pathways at specific points. And then catabolism produces molecules that can also be used for anabolism. So a lot of times when we break down molecules to generate energy, some of the byproducts of the breakdown can also be used for synthesis of other things in the body. Now, one of the last processes we'll talk about is fermentation. Fermentation is an anaerobic process, which means it occurs when there's no oxygen present. Fermentation consists of glycolysis followed by a reduction of pyruvate. Glycolysis is used by virtually every organism on Earth. Early forms of life appeared under the anaerobic, no oxygen conditions existing before photosynthesis helped to generate oxygen in the atmosphere. Some microbes lack energy for cellular respiration and rely solely on fermentation. Various organisms still thrive in places where oxygen is limited or absent, such as in the stomach and intestines of animals, deep in the soil, bogs, and marshes. Fermentation allows NAD+ to be recycled when oxygen is absent. If oxygen is not available, the second stage of glucose breakdown is fermentation. Fermentation does not produce any ATP. In fermentation, pyruvate remains in the cytoplasm and is converted to either lactic acid, ethanol, or carbon dioxide. Under anaerobic conditions with no oxygen to allow the electron transport chain to function, the cell must regenerate the NAD for glycolysis using fermentation. So, when there's no oxygen present, glucose can be broken down to pyruvate using glycolysis, which will generate 2 ATP. and then fermentation follows solely in order to regenerate the NAD necessary for glycolysis to continue. If the supply of NAD were to be exhausted, glycolysis would stop and energy production would cease and the organism would rapidly die. Organisms use one of two types of fermentation to regenerate NAD+. Lactic acid fermentation, which produces lactic acid, or alcohol fermentation, which produces alcohol as a byproduct. So keep in mind that oftentimes when books say fermentation generates 2 molecules of ATP, it is actually the glycolysis that precedes the fermentation that is producing the 2 ATP molecules. And then the fermentation will follow glycolysis when there's no oxygen present in order to regenerate the NAD+ that is needed during glycolysis. So some examples of where you might see lactic acid fermentation. This is in the muscles of animals. And a lot of organisms such as yeast and bacteria will do alcohol fermentation. So as mentioned, some cells will ferment pyruvate to form lactic acid. Lactic acid formation produces lactic acid from pyruvate. Muscles that are working hard enough to use up all of the available oxygen in the tissue ferment pyruvate to lactic acid. To regenerate NAD, muscle cells ferment pyruvate to lactate using electrons from NADH and hydrogen ions. A variety of microorganisms will use lactic acid fermentation, including the bacteria that convert milk into yogurt, sour cream, and cheese. Human beings will often do fermentation in their muscle tissues, such as when they are doing strenuous activity and oxygen is not getting in fast enough for the muscles to function. So a lot of times what will happen is the muscle cells will do glycolysis, which produces 2 ATP, enough to keep the muscles moving, and then the fermentation will follow in order to regenerate NAD in order to keep the glycolysis going. However, while fermentation in humans can occur in the muscles for a short-term burst of ATP energy, it is not sufficient in order to keep the whole organism functioning for very long. Now, some cells will ferment pyruvate to form alcohol and carbon dioxide. Many microorganisms such as yeast engage in what we call alcohol fermentation under anaerobic conditions. This generates alcohol and carbon dioxide from pyruvate. As in lactic acid fermentation, the NAD+ must be regenerated to allow glycolysis to continue. During alcohol fermentation, hydrogen and electrons from NADH are used to convert pyruvate into ethanol and carbon dioxide. This releases NAD+, which can accept more high energy electrons during glycolysis. So oftentimes yeast, when we bake bread, the reason the bread rises is because yeast will undergo alcohol fermentation after glycolysis, and then the gases that are given off during this fermentation are what makes the bread rise. And also sometimes if you notice when baking bread with yeast, you will smell what smells like alcohol as the yeast are fermenting during this process. So some disadvantages and advantages of fermentation. Fermentation is a quick way to make ATP without oxygen. And remember, it's not exactly the fermentation making the ATP. Glycolysis makes the ATP, but the fermentation produces the NAD necessary for the glycolysis to continue to make the ATP when oxygen is not present. Some of the byproducts of fermentation are toxic to cells. And fermentation after glycolysis does not produce as much ATP as cellular respiration following glycolysis. This concludes our discussion of cellular respiration and fermentation.